will start out here with just a few introductory thoughts about modeling in general. So I think we all are aware of that computer models change the world. So today you would never dream about building a bridge without doing the math. We can calculate if the bridge will hold or not. The same goes for a lot of mechanical things. You would not simply just do trial and error and figure out how to make a car. We know how to do it. We can calculate that. The same thing will happen in biology. The next revolution possibly is in that field, but we are very far from achieving this. Here you see adrenaline bound to the adrenergic receptors with waters moving around in a molecular dynamic simulations. And we can run these simulations, but they're still not very predictive. And we can still not design a molecule that does exactly what we do. It's still trial and error. But it will happen. It's just a question of when. So our research group works in the field of computational, structural, and chemical biology. We focus on two major types of proteins. One is G-protein coupled receptors, where we've done the majority of our work, but I've become increasingly interested in enzymes, actually not for many reasons, but one big reason is actually that it's easier to get structures. And I love structural biology, so then I get to see more compounds in complex with proteins. We use a diverse set of methods, ranging from molecular docking to molecular dynamics, also protein structure prediction, before, of course, with homology modeling, and now increasingly with AI methods. And of course, AI methods come into everything that we do today. But the basis is usually a molecular mechanics force field. We try to calculate through interactions why a molecule might work or not. We use this to study protein function and the molecular mechanism of protein function. And all of these projects usually involve modeling a protein ligand complex. And once we think we can do that, we can also use these approaches to discover novel ligands. So our hope is that structure-based virtual screening, which is the main focus of this presentation, will be able to contribute to drug discovery. We know that when you screen large libraries, doesn't matter if it's HTS or DNA-encoded libraries, most compounds will not work. If we were able to predict the interaction between a ligand and a protein, maybe we could calculate that. So our approach has been to try to develop different strategies and then connect that to an actual experimental project. We believe that it's most useful to learn by doing. That's why we design compounds, we make them, we test them, and at the same time maybe come up with ideas on how to accelerate the drug discovery project, but also find leads for drug discovery. And we mainly use the program DOC, which is developed at the University of California. So the question that we've been asking ourselves during the last years is, how can we conquer the chemical space? So the needle in the haystack problem is very challenging. There may be 10 to the power of 60 different drug-like molecules, and we can sample many of them with molecular docking. And there's been a revolution in the field. Before we had access to millions. Later now we got access to billions, soon maybe even trillions of molecules. For example, through the company Enzyme that can make compounds on demand. I can order them and get into my lab for reasonable cost in just a few weeks. But the challenge is in how do we optimally search through this space? And I will show you 3 case studies or 3 types of approaches that you could use to conquer the chemical space. The first one I call brute force. It's basically, why don't we just evaluate all of them? It's what we used to do, and we can still do for hundreds of millions to billions of compounds using DOC. The second approach is artificial intelligence. So we can use AI to try to figure out what a compound should look like. For example, you could do it through generative model, but you could also use AI to try to pick out the best compounds inside the virtual library. And those could contain billions or even larger number of compounds. The last approach I decided here to call RI, real intelligence. And it actually, in this time, refers to a fragment-based approach. With fragment-based approaches, you build a molecule from a very small one into a larger one, and that allows you to explore space very efficiently, as I will show you along the way. So basically, one case study, or maybe in the first case, 2 case studies on how we have successfully used this. The first story begins, it's the oldest one, and begins around the beginning of the pandemic, where we got the opportunity, as many others in the world, to try to find inhibitors of different drug targets. And this work was carried out by a lot of people, which I can't have time to thank here, but the 2 people from my lab were Andreas and Dewey, a computational and medicinal chemist. So if we had the antiviral in the beginning of the pandemic, lots of lives, lots of grandmothers, grandfathers would have been saved. And today we have a drug, Paxlovid, but it has limitations. So although the pandemic is over, there's still interest and need to develop new inhibitors. And I will take you through our story, which started around 2020. So at that point, the pandemic started and the researchers started to identify drug targets, and we became interested in the main protease, which cuts up polypeptides. So it's crucial for viral replication. And we only knew of one inhibitor, which was compound X77. And here you can see the crystal structure of that compound. It's fairly big, and but not very potent, and has crappy cellular activity. So by no means could this be used as a drug. So we decided to try 2 different screening strategies. One of them we called ultra-large virtual screening, in which we tried to fit in hundreds of millions of compounds into the binding site. And these are lead-like molecules that should have decent potency. But at the same time, there were several studies looking into fragment-based drug discovery. Sorry, I'll just open that again. Yeah, so there were several studies into fragment-based virtual screening where we could use starting points identified by crystallography and then try to grow a molecule into another pocket. So this is the ultra-large virtual screen where we screen 235 million compounds with over 200 trillion complexes scored. So this we do on the national supercomputer centers, and you can see here lots of different diverse compounds once I get going. And here you see just the molecules that fits into the binding site of this enzyme. These were then synthesized by Enzyme, about 100 molecules. When we tested those experimentally, we could see that there were 3 compounds that actually showed activity both in direct binding assays with SPR and through enzyme inhibition, but they were not very potent. You can see here that the best ones maybe are around 30 to 50 micromolar. But we could confirm them in the binding assay, and we also got crystal structures of 2 compounds, giving us the confidence to optimize them. And here you can see the best compound, again, not very impressive, around 40 or 23 micromolar. But when we solved the structure, we could see that the docking model agreed very well with the experiment, almost a perfect overlay. So we're thinking, okay, this protein we have under control. We can perform good predictions. The crystallographic screen, there we took advantage of the massive efforts at the diamond light source, where they solved crystal structures together with fragments. So in total, they screened more than 1,000 fragments very early in the pandemic and identified active site fragments that they shared with the whole research community. You might be familiar with the COVID moonshot effort, in which these were used as a starting point for drug discovery. In this case, we tested the fragment, one of them that we liked. It was very weak. And then we identified vectors or ways that we could grow these fragments and searched into the chemical libraries. And we found around 2 million compounds that we thought were relevant. Then we docked these with a constrained docking method. And here again, you can see the binding site. And I just want to show you some of the fragments that we found in the structure. So here you can see some fragments that the diamond light source found were binding to the active site. None of them show any inhibitory activity, but it gives us an idea of how we could grow things. And here you see one of my favorite fragments. There's another one, and I will go back to my favorite fragment here very soon and show you how many compounds you can find in the enamine libraries that are superstructures of these. So there you have the fragment, we do a simple substructure search, and here you see different compounds. And when we dock these into the site, we constrain the docking so that we can grow into a specific site. And of these, we synthesize 93 compounds with the help of enamine. Of these, we got a similar hit rate. About 5 compounds were confirmed in both binding and enzymatic assay, a little bit more potent than most proton fragments compared to the lead-like molecules. And we also solved crystal structures, but you can see that many of these compounds belong to the same scaffold. So basically, one crystal structure would confirm that we had the right structure. And here you see IC50 values, KD values, so they're binding, but not with very high affinity. And we could see that the experimental and docking model again agreed very well. This was maybe not very difficult because we knew how the initial fragment was working, but we could improve it. So if we compare the 2 screening strategies that we did, of the ultra-large screen, hundreds of millions, to the fragment-based approach, which used experimental information already, we could see that the results were similar. We got about 3% or 5% hit rate, about the same. The compounds were a little bit more potent with the fragment-based approach, but still comparable. And when we look at the number of scaffolds, we found more scaffolds, of course, with the ultra-large virtual screen since it used a diverse library. So I was a little bit surprised by this because I thought that the ultra-large library would perform less good because all of this diversity is hard to score for the scoring function. But both approaches give weak inhibitors that we could use as starting point to optimize. So our next question was then, could we use the structures, the chemical structures that we identified to optimize the compounds, again using only commercial chemical space such as the enamine database. And here you can see some of our optimization efforts. So in the library, we focused on hydantoins, and there were 43 million compounds to select from, like a fantastic buffet. And we docked those, we selected some, and you can see here that we go for about 18 to 1 micromolar. We optimized also a second part of the molecule. We're again able to optimize potency, but we end up around there, around the micromolar potency, where we have to go to in-house chemistry. So there, our in-house chemists combined the 2 substituents and managed to find a compound that was even more potent, then reaching the sub-micromolar level. So this is how we work in all of our projects. Initially, we focus only on the make-on-demand libraries where we have billions of options. And once we feel we're onto something, we involve our in-house chemist. So here you can see one of our favorite compounds that we synthesized later, about 400 nanomolar molecule. And you can see the crystal structure. And using iterative crystallography, where we continuously could solve structures, we could also optimize this molecule. So here you see the final compound in the published paper, which we call compound 19, which is 77 nanomolar, high affinity also by SPR, and low molecular weight. So compared to other available compounds, this was a very efficient molecule that we had developed. So we were curious, of course, if they could have cell activity. So we went on with testing in cell-based assays. And here we compare a couple of different compounds. We compare to GC376 and ML188, which were compounds that were available at the beginning of the pandemic. And as you can see, our compound in these cell-based assays to see if we can reduce the amount of virus or cytopathic effect, you can see that our compound is substantially more potent. This is not only because the compound is more potent by itself on the target, but also because it has better ADME properties. It gets into the cell, doesn't get degraded too quickly. The other compound we compare to is the compound that later became a drug, the Paxlovid, and we can see that we compared about comparable with that in cell assays. So we were really, this was a very good project for us because we got good resources to run the project, test lots of compounds, and we could basically develop a quite potent molecule in about 12 to 18 months. This is project also ongoing, and our even more optimized compound was recently evaluated in a hamster model with good effect. I also want to show just one brute force slides on GPCRs, because we've had even more success there for receptors. Here's one example for orphan receptor GPR139, which is a target for schizophrenia. We recently got the first cryo-EM of this orphan. Nobody really knows what activates it, so it's super interesting protein. So we screened 235 million molecules. We selected among the very top ranked and synthesized 68 candidates. And among those, we found, I think, 4 to 5 ligands, continued optimization, initially almost exclusively inside the commercial chemical space. And we identified super potent compounds, I think double-digit nanomolar. We saw the structure of the receptor, which you can see here, which confirms the experimental, the computational prediction. It agrees well with the experimental. And one of these agonists that we found is one of the most potent ever for these targets. And it also shows in vivo activity in a behavioral model. So it's really nice how we now, as computational chemists, actually can move from nothing to a potent compound with in vivo activity in a fairly short time, using only commercial chemical libraries such as enamines. In the second part of my talk, I want to talk about machine learning. It's the second approach that we have become interested in. And here it's Andreas in my lab, Israel, and Leonard that worked on this for a number of years together with machine learning expert Ulf Norinder, and also had some experimental collaborators. So the challenge here is that we dock all of these compounds in brute force, but many of them suck. Like, they don't get good scores. We would never test them. So what if we could improve the enrichment of good molecules? So we only screen a small part of the library, and that's basically the blue ones, the ones we really want. And here you see the distribution for one of my favorite receptors, the A2A adenosine receptor. And as you can see, yeah, lots of bad molecules and just a small fraction of good ones. So we decided to try to develop a machine learning protocol where we screen a small number of compounds, and then we learn from that and predict a new set, thinking that we could maybe identify the best compounds in the library. So instead of docking 235 million molecules, maybe we can just dock 5 and get the same result. So we first docked the training set, then we train a classifier, and we tested many different options, deep learning, gradient boosting, and finally we ended up with Cat Boost. It's not a deep learning method, but it performs as faster than the others and gives the same results. Then we used an approach called conformal prediction to classify them into either actives or inactives. So like which set we should test. And this then results in predictions. So instead of docking 235, we dock about 6 million compounds. And we benchmarked this a little bit. I would just show some results. So it's clear that there is a target dependence. Sometimes this works great and sometimes not. For example, for the main protease, the target of the first part of the presentation, it worked amazingly bad, not very good. Whereas the A2A adenosine receptor that I showed in the previous slide, it worked quite well. But for many targets, you can find 90% of the compounds that are good by only screening a small fraction. And we know this because we actually docked all of them also as a control. So here you can see an example for the A2A adenosine receptor, the recall of the top 10,000 molecules. And you can see here, if we increase the training set from around 50K to 1 million, you can see that you can find 90 or almost all of the compounds in the first 10% of the machine learning evaluated. But basically, you could speed things up enormously with this approach. And we chose 1 million for training set, but you can clearly see here that you could do with less. It's just because it's fairly easy for us to screen 1 million compounds. It's not a very big effort. We could also see that if we ask the machine learning model how confident it was in the predictions, we could shift the curve even more. So that means that if we say, okay, why don't you just give us the most confident molecules, then they have a much lower energy than if we grab all of them, that the conformal predictor would ask us to evaluate. And this is important because even if you can reduce the library to 1% of the total library, if you go to billions or trillions, that's still a lot of molecules. So we need also to be able to rank molecules by their confidence. But the elephant in the room is that these are just predicted scores. There are no experiments involved. So we, of course, also wanted to run some experiments. So we took the D2 dopamine receptor, target for Parkinson's disease, and screened first 1 million molecules as a training set, and then we evaluate that on 3.5 billion molecules. And that's basically all lead-like molecules in the NME library. Then we took 5 million predicted ones and docked those, and as you can see, they have dramatically better scores. And we tested about 31 molecules from enamine, and we found that 2 of those worked. So 2, so that's I had expected better, right? So we screened a much larger chemical space, and the hit rate is about the same as we've had with smaller libraries, but we're still reaching this enormous chemical space. So maybe we need better scoring functions, but certainly we can find all the best molecules with the docking scoring function very rapidly. So this made me think about, what do I think the machine learning and the large libraries can help us with? Well, the smaller libraries seems to be able to give us starting points for many molecules, for many targets. We can find ligands that bind into them. But when we add different constraints, and that could be a certain ADME property, it should cross the Blaine-Brown barrier, it should be selective. The more properties you add, the smaller the chemical space. So whereas you might have a ligand in an HTS set of only 10,000 molecules, if you also wanted to have good ADME properties, there will be none in the library. So the bigger libraries, which we can then screen lots of different targets with machine learning, there we can address questions like polypharmacology. Can we find molecules that binds to 2 unrelated targets? And here I show you an example of that, where we screen the A2A adenosine receptor and the D2 receptor. So in the training set, you can see that there are very few molecules that have a good docking score for both targets. And that's because they're quite different in structure. But if we train a machine learning model to predict which molecules could bind to 2 targets, as you can see the red dot, where the majority of the compounds are, moves. So you can enrich enormously the number of compounds fulfilling several different properties. And the cool thing about all of this is that this is a free tool. You can download it. It will be 1,000-fold faster than if you explicitly screen every molecule. We've already validated for several receptors. It's easy to use, and you can use it with any docking program. You don't have to use doc. Use whatever you want, train it, and see if you can get compounds with better scores. The final story of today is I call the real intelligence part. And it's because I like fragment-based drug discovery so much, and the sort of intellectual challenge of working with fragments. And this is a story that we also published this year, and it was Andreas and Dewey on the CompChem side, and lots of great experimental collaborators. The target in this study, which I won't talk about so much, we call it OG1, and it's involved in DNA repair. So the interest from our side comes in treatment of cancer, and it's also possibly a target for inflammation. It's also interesting because it's a DNA binding target. So it's actually has a challenging binding site. And if you look at the different structure, you can see that it's very flexible. The available inhibitors, and you see the one here in the screen, was decent. It was like 300 nanomolar, so not really a drug candidate, but it had pretty poor pharmacokinetic properties. So we wanted to see if we could use molecular docking to improve on the scaffold that they already had, and in particular find a new scaffold that they haven't seen before. So we tried it. In this case, we also tried 2 different strategies. So the first one was what we call then the ultra large library, There, just as before, we take hundreds of millions of molecules, we dock them into the site, and we hope to find a potent compound. In the case of the main protease, we found several compounds with high micromolar potencies. The second approach, there for the MPro target, we use first the crystallographic screen. But what we really want is to first dock all commercial fragments. So we dock lots of small fragments, less than maybe 19 heavy atoms or 250 Dalton. And there we hope to identify fragments, and in a second step, optimize them to potent leads. And it's not obvious which of these would be the most efficient strategy. You could, for example, imagine that the hit rate could be higher from the fragment screening, as they usually is. And then you could test less molecules in the beginning and then put more in the optimization phase. Or you could just go for the bullseye, right? Let's buy 200 compounds from the large library, and hopefully one of those will be very potent. So we started out with 2 screens, actually. We performed these in parallel. We did first the lead-like docking, where we screened 235 million molecules. These molecules essentially fit the entire binding site. So they go from the bottom of a buried site, reaching out into the solvent. And there we selected 65 compounds, which were tested in our thermal stability assay. But unfortunately, there were 0 hits from this screen. We tested them at 99 micromolar, so hoping that they would at least be micromolar compounds, but it didn't work. So here we can see a contrast from the screen we performed for the main protease, where this gave us lots of starting points, whereas in this case it did not. So the next step was to perform a large-scale fragment docking. So when we do experimental fragment screening, we screen around like 1,000 molecules maybe, or even a few hundred. But here we could dock every commercially available fragment, which was 14 million. So we sampled these in the site and focused in particular on the buried pocket, which we thought looked like a good starting point. And then we selected 29 fragments, about half as many as we did for the lead-like set. And there we found compounds. 2 of them, we had sufficient stabilization at around 500 micromolar to be visible, and the other ones we discovered through also testing them in the crystallographic screen. And you see they're quite diverse. They have this central amide, but sample new chemical groups on each side. So we thought that was very interesting. Then we solved the structures of these, and we could see that for a majority, perhaps all except one, the binding post predicted by docking was correct. Their overall interactions were captured, and then some groups were flipped here, as you can see, for compound 2, but overall the prediction was accurate. And using the crystal structures, we tried then to identify how we could optimize these molecules, and we were mostly interested in the first compound here, because it had the largest stabilization. You could also think about maybe one should go for the one with the highest number of analogs. That's also another strategy you could use, because to be honest, they're all crappy molecules, but you should work on those which you can optimize rapidly. And you can see here that I also marked a reference inhibitor so that you can see approximately in space where they overlap. Then we perform optimization, and we start, as always, in the enamine library, and we define kind of like a vector. So we make smart searches, and we find 42,000 possible molecules that contain the motif we think is important for this fragment. We synthesize those at enamine, and you can see there's an improvement both in the IC50 and the temperature shift, and the molecule has grown slightly, added an aromatic ring. And then in the next step, we continue to grow this molecule. But as we make them more complex, of course, there are fewer molecules in the library. But we could get to 6.6 micromolar. And here you see that we moved into another pocket. And this was actually possible by iteratively solving crystal structures. So this pocket that we see here was actually not open when we solved the first structures, but as each time we grow the molecule, we try to solve a new structure, giving us the possibility to take into account induced fit, because usually we use rigid structures. But then we couldn't find relevant compounds in the enamine library, and again turned to the in-house chemistry, which always is a close collaboration between the computational chemist and what I would call the real chemist that makes the molecules. So here we docked thousands of analogs just based on commercially available building blocks, and we synthesized. Here you can see the crystal structure of the parent compound, and then we use that structure to identify molecules. In particular, we wanted to reach a new hydrogen bond with an aspartic acid. And we managed to do that. So this hydrogen bond, actually, it resulted in around 10-fold improvement of potency. And that was basically the final compound of the project. So if we look at the efficiency of our project, we could see that whereas the other compound had been discovered through HTS and extensive optimization, I think more than close to 20,000 compounds had been tested to find it and published by our colleagues in Science. We found this molecule, the new scaffold, by testing only about 100 compounds. We have to do a lot of computation and be strategic about how to use it, but we can truly go from nothing, which you can see the fragment here in the curve, to something in a quite short time and testing quite few molecules. I just want to show you a little bit of biology also, because we tested it for selectivity. We can see that it's selective against most DNA glycosylases that we worry about, not NTH1, but overall very good selectivity. We also tested cellular activity in CETSA and also laser-induced recruitment, so we could see that we could inhibit that. And we also tested it in an inflammation-relevant assay. In all of these, our compound performed comparable to the compound that was previously discovered. So now we go over to a more philosophical side. So what could we learn? What strategies could you use to explore the chemical space? I've been very inspired by the Ramon Group's work on the GDB and the fact that you can enumerate all possible chemicals up to a certain number of heavy atoms. So here's, for example, GDB 13 with a certain number of elements and some constraints for stability. They get up to around 1 billion molecules. And you can extrapolate their data and get like, maybe there's around 10 to the power 33 to 10 to the power 60 drug-like molecules. So Andreas, in my group, he developed what we call the universe generator. And that enumerates all fragment superstructures. So it allowed us to look at the molecules that we've discovered. How many superstructures do they really have? And how many can we reach today? And I won't get into the details of it, but you can download and run the code yourself. But this is like a, this is what we can say about the scaffold that we worked on. So if you look at the core fragment of our compound, there are about 10 to the power of 6 physically available molecules. Somewhere in the world you can buy about 1 million compounds that are fragments, like if they're less than 13 heavy atoms. The theoretical possible is 1,000 times more. So we're doing okay, but there's still a big gap. But in the superstructure space, which we allowed in lead-like molecules, we're missing an enormous number of compounds. So there's about 10 to the power of 13 possible superstructures of this molecule. So that led me to do another thought experiment on what is the most efficient way to explore chemical space. And I'm going to reach the same conclusion as many others have done before me. Fragment-based drug discovery can't be beat. That's the bottom line. But let's go through the math. So if you have a 10 to the power of 22 space, and you want to find this particular molecule, one of these 10 to the power of 2 is this nice scaffold. How can we find it? One way is to start by doing a fragment screen. And there are about 10 to the power of 9 then molecules of heavy atom 13, around that number. That's typical of a fragment in a fragment screen. When we've done that screen, if we can identify this compound and solve a structure, we could identify all the superstructures. And those I discussed on the previous slide. And if you, in addition, take into account the structure, crystal structure, so that you don't elaborate vectors that seem unproductive, they would clash with the protein, you get to about 10 to the power of 9 molecules. So these are, we enumerated all these molecules. So it's an enormous number, but still in total, it's only 2 billion. Today on the supercomputer center, we dock 2 billion molecules. So if we use a fragment-based approach, we can enumerate first all possible fragments, and then we can enumerate for one of those fragments all relevant superstructures, not just the commercially available or synthesizable, all of them. And that's pretty cool. But then we can compare, what if you do as we've done? You take just a random sample or the enamine database and screen 2 billion molecules. If you do the math, this random sample of 2 billion in this space will not even contain one example of the scaffold that I show you in the middle. So the diverse screens of billions of molecules are very limited in the chemical space. And the only way maybe we can win is to start from a fragment-based approach. That's the end of the philosophy part of the presentation, and a little summary. So I've showed 3 different approaches: brute force, artificial intelligence, and real intelligence, the fragment-based approach. I think they're complementary. We can see and cannot really predict why some work in one case, some work in others. So use them in parallel. But you need to be careful, because the choice of approach depends on many experimental factors. For many targets, fragment screening is not suitable because you don't have a sensitive assay or access to structural biology. So the first thing one should look at when running a project is what assays you have. We see that the commercial libraries are large enough to find starting points, both receptors and enzymes. For enzymes, we find weak compounds, micromolar. For GPCRs, which many have nice small pockets, sometimes nanomolar. But when we optimize, we often at the end, even if there's 76 billion compounds available, in the end, we have to go into in-house chemistry. It seems we can't avoid that yet. The brute force docking is becoming more difficult. You know, now there's tens of billions of molecules that we could dock in the lead-like space, and we simply don't have the disk to do it. So in the future, the AI-accelerated fragment/symptom-based approaches will need to solve the problem. And in all of these questions, I think we're in urgent need of scoring functions, both to improve the initial hit rate. So, you know, I've been bragging here about discovering molecules, but the truth is about 95% of the things that we predict are wrong. 5 to 1% of the molecules works. It's ridiculous. And so we need better scoring functions. And in the optimization efforts, in particular as they get expensive, we need more methods there in order to guide the right substitutions. So far, we've not been able to make methods work, such as FEP, to really be used in a prospective setting. So we need more work there. And that's the end of my presentation. I want to thank the people contributing, Andreas, Dewey, Israel, Noor, Flavio. Andreas now has his own lab after a postdoc at MIT. I want to thank collaborating groups. There's tons of them. We would be nothing without them. And we're funded by different funding agencies. Thanks for that. And I want to thank you for attending, and happy to take questions.